My name is Jacqueline Wahura, and I'll present to you part of my PhD work on the changes in the Anopheles arabiensis transcriptome and gut microbiota associated with the endosymbiotic Microsporidia MB. Microsporidia MB is an endosymbiont found in Anopheles mosquitoes and was shown to impair the transmission of Plasmodium falciparum. It is vertically and horizontally transmitted, ensuring its stable transmission in mosquito populations. However, the mechanism it uses to impair the transmission of Plasmodium is not known. In this study, we endeavored to determine whether mosquito infection with the endosymbiont activates the host immune system and whether it modulates the gut microbiota to enhance its transmission blocking phenotype as a step towards unraveling the mechanistic basis of Plasmodium transmission blocking using this endosymbiont. Transcriptomics data in this study was generated using the Oxford Nanopore Technology MinION device and was analyzed using a metatranscriptomics approach as listed in section 1 of the methods section. Gut microbiota data, on the other hand, was generated using the Illumina MySeq platform by targeting the V3, V4 region and analysis done as highlighted in part 2 of the methods section. We observed that Microsporidia MB infected mosquitoes recorded a down-regulated immunity and an upregulation of juvenile hormone synthesis in non-blood-fed mosquitoes. Juvenile hormone biosynthesis upregulation was previously associated with a down-regulated immunity and an increase in bacterial density, and this could be suggestive of a unique microbiota structure in Microsporidia positive mosquitoes. In congruence to the study, we observed a relatively low abundance of Serratia and Pseudomonas in Microsporidia MB positive mosquitoes, and the two bacteria have been associated with immune system activation. This suggests that their downregulation in this particular data set was associated with downregulated immunity as observed in the transcriptomics data. In addition, we observed an increased bacterial density in Microsporidia MB positive mosquitoes as compared to the negative mosquitoes, and this could be associated with the downregulation of the immune system. Lastly, we observed a distinct bacterial clustering pattern in Microsporidia MB positive mosquitoes in blue compared to the negative mosquitoes in red. In a nutshell, this dataset suggests that Microsporidia MB infection in mosquitoes modulates the mosquito physiology through a complex interplay of immune suppression, metabolism, and gut microbiota restructuring in the non-blood-fed mosquitoes. Gene expression and gene ontology analysis 24 hours post-blood meal indicated an upregulation of immune system and downregulation of metabolic processes. A key gene that was upregulated is the lipopolysaccharide tumor necrosis factor, which has been associated with hemocyte differentiation given Plasmodium infection. This suggests that there is a role of cellular and humoral immune responses in Microsporidia MB infected mosquitoes as far as Plasmodium transmission blocking is concerned. In addition, we observed a proliferation of microbes with immune system activation roles, such as Serratia and Pseudomonas, and a distinct clustering in microbes indicating that MB infected mosquitoes and negative mosquitoes had a distinct gut microbiota structure, and this could be favoring the Plasmodium transmission blocking phenotype. In a nutshell, Microsporidia MB infection modulated the immune system 24 hours post-blood meal as well as the gut microbiota, and this could actually be the strategy that the endosymbiont is using to impair the transmission of Plasmodium. 48 hours post-blood meal, we observed that the immune system was activated, while some factors associated with immunity were also down-regulated, indicating that immune system activation was at the point of inflection at this particular time point. In addition, Serratia and Pseudomonas, initially proliferating with blood feeding, had relatively low abundances at the 48-hour time point, similar to what was observed with the non-blood fed mosquitoes, and this could be playing a role towards the waning of immunity at this particular time point. In addition, we observed a distinct clustering pattern in MB positive mosquitoes as compared to the negative mosquitoes. In a nutshell, this dataset suggests that Microsporidia MB positive mosquitoes experience a wane in immunity activation 48 hours post-blood meal, and this suggests that Plasmodium transmission blocking in MB-infected mosquitoes is more pronounced early, that is 24 hours post-blood meal, and tapers off by 48 hours after blood meal. In a nutshell, it is evident that Microsporidia MB plays a role in modulating the gut microbiota, and it does this by favoring microbes that have a Plasmodium transmission blocking phenotype, specifically those associated with the immune system activation. In addition, we've observed that Microsporidia MB affects factors that are associated with hemocyte differentiation, indicating that Microsporidia MB impairs the transmission of Plasmodium by activating both the cellular and humoral immune responses. The findings from this study form a basis of elucidating the intricate mechanism that Microsporidia MB uses to block the transmission of Plasmodium and will be harnessed in developing a scalable and sustainable Microsporidia MB strategy. Thank you.